Well, thank you, Dominic. It's a pleasure to be here, and congratulations on having these annual meetings. I know they're a great deal of work. They're very much appreciated, and it's a good way for people to get together and change and discuss and disseminate ideas. It's always a pleasure. Thank you. I've never worked with CAR Ts or MSCs, and the field of stem cells has gotten so large that, I just stay focused. And originally, hematopoietic transplant for autoimmune diseases, I still treat those patients, but my work has shifted over the last decade to embryonic stem cells and IPS cells. And because of patents that we have, and we have 7 patents on our cellular technology, we set up a company called Janani, and you can see our corporate logo up here, right there at the top left, Janani. And we've also trademarked that logo, and we've trademarked our logo here, Engineering Regeneration. And so I just want to be upfront about that in case there's a conflict of interest, but we do not yet have a product that we can use in a clinical trial. So I think, it shouldn't be much of a conflict of interest here. We are developing our first clinical protocol, clinical for humans, and hopefully, I don't want to jinx myself, but I hope to be able to have that protocol finished this year. So what I'm going to talk about is our IPS technology, and it's a new word that we coined ourselves, and it's in our patents, and we call it transbiosis. And transbiosis is a neologism for transfer of life. It literally, biology or is Greek for the study of life. So transbiosis, or bio is life, is a transfer of life. We've done it with embryonic stem cells, and we've also done it with induced pluripotent stem cells, or IPS, which is a relief for me because there are ethical issues that surround embryonic stem cells, and doing new things, you generate controversy, and I just don't want to go down that road. However, in the lab, we do work with mouse, monkey, rhesus macaque monkey, and human embryonic stem cells. But we also have human-induced pluripotent stem cells that we generated ourselves from fetal foreskin. So it's a simple concept, transbiosis. Cells help cells. And this goes back to parabiosis. And there's a publication by a UK physician, Dr. Burt. He was a surgeon, published in 1863. And what he did is he surgically connected the circulation of a young mouse to an older mouse, and it made the older mouse younger. And that was then kind of forgot for a while, but recently there have been a lot of publications in the late 1900s and 2000s confirming this at a molecular level out of many institutions, including Stanford, with publications in Nature Medicine and so forth, on sharing the circulation of a young mouse with an older one, making the phenotype and gene expression profile of the heart younger. And so it's been held up that does happen. The exact mechanisms occurring are still not clear. And then there's a concept, and that was Dr. Avery in 1944 from the Rockefeller Center. And it was known that bacteria could develop resistance to antibiotics, but what they discovered is that dead bacteria transferred antibiotic resistance to live bacteria. So you take bacteria, kill them, and they would transfer their phenotype, their characteristics, to the living bacteria, even though they were dead. And they called that transforming factor, and that was repeated by several publications in the 1940s. What Avery did is he took the bacteria and he added a soap and salt, which broke open the membrane of the bacteria, and to that he added isopropyl alcohol, which caused the DNA to precipitate out of solution, and you stir it up and you get these long strings of gooey white DNA, and you can actually pick it up and it'll be like a string, gooey string over your finger hanging down. You can do that in your kitchen with a tomato or a strawberry and just grind it up and filter it and then add soap and isopropyl alcohol, stir it, and you'll see the white stringy DNA pull out and take it up. You can lift it up. You can have someone's DNA in your hand. It's kind of interesting. And he took that and showed that that contained the transforming factor that live bacteria would take up and give them antibiotic resistance, even though they've never seen that antibiotic. So that transforming factor became known as DNA or genes. But it's interesting that a live cell will take up material from a dead cell and change its genetics and change its phenotype and genotype. And then feeder layers came about, and that was by Puck and Marcus in 1955. And to do research, you need to grow cells in a lab, and a lot of cells just won't grow in a lab. And what they found is if you put the cells over another feeder layer of cells, a feeder layer that won't grow because you've like irradiated them, so they cannot grow, but they're not dead. They won't divide, so they're not going to run over the plate, take it over. You can put other cells on there and they will grow and they will divide and you can passage them. So this feeder layer is used today a lot. It's done all the time in labs, and it allows you to grow cells easily, and all the factors provided aren't always clear, but cells help cells. And then transdifferentiation. It was, an adult cell, they found that if you use a stem cell, and it can be a hematopoietic stem cell or mesenchymal stem cells or even a primitive early embryonic stem cell, that it will improve the function of damaged tissue. It does that. And there was this speculation in the literature in the late 1900s, early 2000s, that there was transdifferentiation of changing one cell type to another. That's not how it worked. It did help the cells, but it didn't cause transdifferentiation in vivo. Nevertheless, you can do transdifferentiation ex vivo, where you can change one adult cell into another type of cell. That's been done all the time, and that really first came out with Davies in 1997. And perhaps, and now what we have patented is transbiosis, where we can use irradiated or lyophilized embryonic or IPS cells to improve any tissue function independent of immunologic differences. So it's MHC independent, which is great. It's a universal nurse cell, or if you will, a universal stem cell to repair any damaged tissue. So let's go back to this transdifferentiation, because that's important. We have to make our IPS cell, and it shows how versatile cells are, that, there's this concept you just differentiate and that's it. You can never go back, but in fact you can. And so transdifferentiation, or reversal of a differentiated cell to its embryonic state, does not occur in vivo under normal physiologic conditions. Otherwise, we'd be falling apart all the time. But it does occur ex vivo outside the body. And perhaps the best example of this reprogramming was by Professor Yamanaka, who got the Nobel Prize for that, in which he was able to generate IPS cells, which are embryonic-like or embryonic stem cells from an adult, fully differentiated cell. And so if you look at the embryonic versus induced pluripotent stem cell, the embryonic stem cell is basically an ovum that is fertilized into a morula, and then which happens in the fallopian tube. And as it passes down the fallopian tube and enters in to the uterus, it becomes a blastocyst, which is about day 6 after fertilization. And that blastocyst is a microscopic ball. You can't see it with your eye, but you can see it under a microscope. And the outer layer is already differentiated into what will become placenta. The inner layer are the embryonic stem cells. And those embryonic stem cells, each and every one of them, can form every organ in your body. And in fact, they do form every organ in your body. It implants in the uterus, and then by day 7 or 8, they already disappear. So we all had embryonic stem cells. We were, in fact, basically just embryonic stem cells until shortly after the blastocysts attached to the uterus, and then they differentiate into mesoderm, endoderm, ectoderm, and they're just gone. And it's a good thing they're gone because they're so powerful that they can cause problems. But they end up forming every cell in our body, and that's the hope, is that you can take embryonic stem cells and remake all the tissue. Of course, you have to deal with MHC differences and how to get it to integrate properly in an adult organism. On the other hand, you can take any type of cell, such as a fibroblast or a skin cell or an intestinal cell, and you can transdifferentiate it or genetically reprogram it back to the embryonic state. And classical reprogramming factors are SOX2, OCTA4, NANOG, KLF, and c-MYC, or LIN28, depending which ones you're using. And these genes, if you can get them inside an adult cell, will make an IPS or embryonic stem cell. That you can then differentiate again into any one of these cells. So that's what's going on in the field with IPS and embryonic stem cells. The advantage of IPS is there's no ethical controversy, plus you can take anybody's cell and do it, so you don't have to worry about MHC differences. And so how do you do it? It goes back to the concept of cloning, that you can take. The cloning was first done by Dr. Gordon in the UK. He got the Nobel Prize for it. So he was knighted by the Queen. He's now a Sir, formally knighted. But he took the intestine of a frog and took the nucleus out of that adult intestinal cell and put it into the egg, ovum of a frog, and then created a whole new frog. And so he was able to clone frogs by doing that. And that led to the discovery of mouse embryonic stem cells, and then eventually to the cloning of sheep by Sir Ian Wilmot, who was also knighted by the Queen for cloning the first mammal, Dolly the sheep, if you remember, from an adult nucleus of an adult cell by putting it into the ovum or egg of a female. And that led to Thompson at the University of Wisconsin creating or discovering human embryonic stem cells. And then finally Yamanaka, who got the Nobel Prize for creating IPS cells in mice, and then Thompson created IPS cells in humans. The reprogramming factors that Yamanaka uses are OSKM, 4 genes, almost the same OS, OCTA4, SOX2, but a little different NANOG and LIN here, NL, compared to KLF and c-MYC by Yamanaka. The problem with those is that you had to get them inside the cell, those reprogramming factors, those genes, and so you had to use a virus to get them in, initially a lentivirus, which integrates into the DNA. It was thought they always had to be there, being expressed, but the danger is you don't know where they integrate. It can cause all kinds of problems, lead to a cancer. So they began to use non-integrating viruses like Sendai virus and adenovirus. We have used a Sendai virus. It'll disappear. It's extra chromosomal. It'll disappear in about 10 passages. And then using a plasmid, putting your construct inside an episomal plasmid, which we've also done. And of course, those are extra chromosomal. They don't get into the DNA and they disappear with passage after about 10 passages. And then it's gone on to just using mRNA, microRNA, protein, small molecules. This has really expanded how to take an adult cell, any of your cells right now, and convert it back to the embryonic state. So there are many ways of doing it, and it's become much easier to do. It's actually, the younger you are, the easier it is to do it. That's why we use neonatal cells. There's no controversy. Neonatal foreskin, it was otherwise thrown away. But it can be done in an adult, but the older you are, the harder it is to do, but it does work. And actually, here's how we did it. We got neonatal foreskin, and the parents, of course, were checked for every known adventitious agent, viral agent, whatever. So what are the cells? And then we just grew and made a master cell bank of these fibroblasts. Here, these are classic fibroblasts. And so we'd put them in a fibroblast media. We'd take a vial from a master cell bank, and then we would add our episome containing our cocktail of, which were really Yamanaka's reprogramming factors, and we were successful in getting a license from University of Kyoto that holds the patents on that to use them. And here's the basic technology where after you, if you look, I don't know why it keeps doing this, but you look, about day 8 or 10, you start to see the cells, some of them start to change. For the first week or 10 days, there's nothing going on. You think this isn't working, but then all of a sudden it does work, and you end up, especially by day 18, this colony of very young embryonic stem cells. So the fibroblast has converted the adult fibroblast back to the embryonic state. And there's many ways you can check that, to confirm the identity of a cell as an embryonic stem cell. Here's just an antibody called TRA1 that is a clear visual presentation. But you can take these cells and put them into a blastocyst, and that particular cell will become a part, even though it's different, of that whole new organism. So it is a true embryonic stem cell, these IPS cells. So then we, in our concept of transbiosis, we irradiate or lyophilize them. A lyophilization is kind of like you sublimate them, so it goes from fluid to gas without ever freezing, and you kind of that way vacuum freeze them over very low atmospheric pressure, and you end up with this kind of white powder, which is nice because you can just store it at room temperature, unlike a live cell that you have to have a media with, and you're concerned about how long you can have it before you have to inject it into someone. In fact, these little vials, we have them, we just keep them on the countertop. It looks like cocaine. So I never carry them around because I'm always afraid TSA will pull me over and they're not going to understand what I'm talking about, and they'll be all excited. But in any event, we do mitotically inactivate them, and so they won't divide, and whether they're living or lyophilized. Lyophilization also mitotically inactivates them. But the reason is that we don't want them to form teratomas, because if you take a IPS cell or embryonic stem cell, you inject it into an animal, it will form a teratoma. It'll form every organ of your body, but in a very mixed up collection of cells, a tumor, and you don't want that. So you irradiate them so they can't grow. And again, the traditional approach is you take your reprogramming factors, you make IPS cells, and then you differentiate them into some sort of adult cell. Our approach is we skip all that heavy work of differentiating them into a cardiomyocyte or hepatic cell or whatever, and we just take our cells, we mitotically inactivate them, and we use them as the therapeutic drug in vivo to treat damage. And it works. And so the good news is irradiated, that is non-mitotically active or lyophilized embryonic stem cells or IPS cells are a universal nurse cell that repair damaged tissue. And here's some different types of animal experiments we've done just to show all the different organs we can repair. Most of this work has been done in mice with mouse embryonic stem cells because mice are not expensive and mouse embryonic stem cells are easy to grow. Some of it we did in monkeys with rhesus macaque monkeys with human embryonic stem cells. We do have monkey, but they're actually the hardest to grow and maintain in the lab. Human are easier but more difficult than mouse. And so we were able to repair myocardial infarction, leg ischemia, spinal cord transection, radiation injury, wounds, no matter what the cause of the wound, and improve cognitive ability in the aged. And our most recent patent is we could actually reverse aging and return animals to a more youthful phenotype. That reversing aging will really need to be a different talk because it really gets us into epigenetics. So it turns out when the human DNA was sequenced, the Human Genome Project, the hope was that'll give us all the answers of what these 3 billion nucleotides are doing in human DNA. That was the hope, because that tells us what our genes are that make the proteins that determine who we are. It turns out how much of that 3 billion DNA is used as genes to make proteins? Anybody want to guess what percent? It's 1%. All the rest, what was it doing? It was called junk DNA, but one man's junk is another man's treasure. And it turns out that junk DNA are the regulators that determine how genes are expressed. It also determines the aging of the cell. And that junk DNA is important in evolution. So if you look at bacteria, 50% of a bacteria's genome is junk DNA, 50% are genes to make proteins. When you get to humans, it's only 1% make proteins, 98% is junk DNA, because a human has to differentiate into all these different type of cells. You don't want a neuron making hair. And that is controlled by the junk DNA. And it's turned out that they're discovering a lot of, quote, genetic problems like myotonic dystrophy, which I believe is the most common European genetic cause for weakness. And the gene is totally normal. When you sequence it, there's no mutation. What's causing the problem is junk DNA outside of the gene. So this junk DNA controls our genes, but more importantly, it controls aging. So, but that would be another talk. We're only going to talk about tissue repair today. So, if we look at a mouse and we induce a heart attack, here we're tying the circumflex artery of the mouse, and we let them go through ischemia for 50 minutes, and you can see the ischemic part of the heart actually turn blue while the rest is red. Of course, they're intubated and they're sedated and so forth. And in some of the mice, we would inject in the penumbra before we release the tie our mitotically inactivated IPS or embryonic cells. In others, we would just use the media they were in. And what you could see, if we looked afterwards, a month afterwards, in heart contractility, our controls that didn't get our pluripotent stem cells had a marked decrease in contractility of the heart compared to the ones that either got the live but mitotically inactivated cells or the powdered lyophilized form. And same with diastolic function. You can see diastolic function is much improved at 30 days compared to just an infarct where we didn't give that one-time injection in the penumbra before releasing the occlusion. So that's very encouraging. It was reproducible. Oh, so we did this in mice. We also did this in rhesus macaque monkeys with humans. Humans are genetically very close to monkeys. We are primates. And it worked. It worked very well for them, too. And so, here's the rhesus monkey data as well. So here's the pre-MI cardiac contractility 1 month later versus the pre-MI 1 month later given our inactivated or powdered cells. And if this is also the same thing looking at diastolic function improving in these rhesus macaque monkeys. We did this with a Miller catheter, which measures pressure versus time changes in the heart, which is the best way of looking at contractility and relaxation. So a healthy heart will relax instantaneously, very high change in pressure with respect to time. A failing heart relaxes slowly, and there's not a very rapid change in pressure with time. The differential is really low. So that returns to normal. The others, we did it with MRI to look at infarct size, which was also decreased. And we did these wounds, here's wound repair 2 weeks after treatment without our IPS cells versus 2 weeks after treatment with it. And you can just obviously see we did it photography, digital recording to determine the area for reepithelialization that occurred at 2 weeks. You can see it's markedly better in the mice that were treated with our inactivated IPS cells. And if we looked at survival after total body irradiation, where the dose would totally destroy the bone marrow, so we had to do a bone marrow transplant. If we supplemented the bone marrow transplant so the animal would live with our IPS cells that were irradiated, we needed a lot less bone marrow for them to survive and recover. So it could be helpful for like a Chernobyl nuclear disaster. I mean, if your bone marrow is totally gone, it's not going to recover it. It's not replacing that. But if there are still some stem progenitor cells there, just a few that aren't going to make it, you're not going to live in time for recovery, this will accelerate it and make it come back faster. And when we looked at intelligence, so we took 2-year-old mice. Mice live to be about 2 years old. And we did a water maze test in them. And we compared that to mice that had been treated a week before with our cells. And what we found is that in short-term memory, that is the total percent of tests failed on the training day, our regular old mice, most of them failed compared to those given our cells, depending how we gave them. And if we looked at long-term memory, which was after the training day, the next day we came back and retested them, and we can see that the mice treated with our cells did much better. They retained memory. They learned faster and they retained memory. So we call this our Flowers for Algernon experiment. If you ever read that book or saw the movie Charlie, which was based on that book, because it's capable of making mammals more intelligent. And so the question you may ask is, how does this work? It kind of sounds science fiction-like. I told you cells help cells. I showed you the history of it, but it's kind of been forgotten. But how does it work? So, of course, one of the ways I can ask is, how do you define life? And I would say life is metabolically active or has the potential to be metabolically active. That is, energy is kinetic and potential. And so kinetic energy is the movement, potential is the ability yet to move. And a virus, when it's not inside a cell, is completely inert. It's no more metabolically active than a rock. But a virus has potential to become metabolically active when it's in a cell. So you can be totally inert, sit there and do nothing, have no metabolism, and have the potential for life. Okay? And most drugs actually are either an inert small molecule that we don't identify as life, or as an inert antibody that we don't identify as life, yet they have significant metabolic consequences. And as an aside, the term cell, anybody know where that term comes from? It comes from Latin, cella, which means storeroom. It's the same derivation as our word cellar, where we would store things. It's because a cell stores all the things that make life. Those things, each one by themselves, whether you take a piece of DNA or a protein or RNA, we don't see it as life. But it is the symphony that makes life. All right, so it's all due to electromagnetic differences from polar and van der Waals forces and covalent bonds in these molecules. It really boils down to electromagnetism. So irradiated and lyophilized cells retain potential at a molecular level for to be metabolically active and help another cell. That's what I'm saying. And when we did microRNA analysis, we found that there were a multitude of genes that were very upregulated in these embryonic or IPS cells compared to adult stem cells, such as a mesenchymal stem cell or an adult cell. And the upregulated genes are all genes that really have to do with gene ontogeny, GO pathways that are involved with metabolism and repair. So when we would treat damaged tissue, it would do the same thing. It would upregulate those genes. It would upregulate metabolism and repair, which is exactly what you want to happen. And then the 20 most downregulated GO pathways all have to do with inflammation. It downregulates inflammation, which downregulates fibrosis, which is a good thing. So, I know when I say these pathways, it involves about 500 genes, about 250 that are markedly upregulated, 250 downregulated. And it also involves a whole set of genes involved in epigenetics, that is histone modification and methylation of DNA, which would have to be another talk. But anyway, we can deliver this by sub-Q or intrathecal or intramuscular or intraperitoneal, or even by spray over a wound, or perhaps even intranasal. Oh, here's our lyophilized powder right there. See that? We can leave it on a shelf for a year, take it off, add a little phosphate buffered normal saline, inject it, and it works. So our initial application that we're writing will not be autoimmune, it'll be amyotrophic lateral sclerosis. The reason is, it's very variable. There's a lot of different genes involved in ALS, neurodegeneration of motor neurons, but from diagnosis to death, the average is 3 years. There's an occasional person who will live 20 years, some 6 months, but it's 3 years on average. And nothing really works. Nothing works. So it's an orphan indication for the FDA, because if you have anything that can help, there's nothing out there. It's a lethal disease, short time period before you die. So that's why we want to go there. We think we'll get a more easy steam rolling through the FDA for this protocol, and we started working on it. We will also be working on traumatic wounds, looking for Department of Defense funding, because obviously a medic could use this in the battlefield. Just pop it right into a wound, which would help start immediate repair until you get them to the MASH, Mobile Army Surgical Hospital, or back to a hospital outside of the conflict zone. So in autoimmune diseases down the line, we would work in wound healing. RA and systemic sclerosis are vasculopathies, and you do get non-healing wounds with them. You do get these ulcers in RA, and we think it would work very well there. In multiple sclerosis, we would use it for non-active progressive MS. In progressive MS, no immune-based therapy works. There are no drugs out there that are really helpful. There's some little paper that rituximab helps a little bit in progressive MS. That's because it's progressive with still some immune activity. Once you're just dealing with the pure neurodegenerative phase of MS, which is where it will end up, there's nothing that works because it's not immune mediated, and I think this approach could work very well. But again, our first protocol will be ALS, because I think we can get an orphan indication to get it moving faster. So this is our cell. We trademarked it as my cell. I am a Fulbright scholar for Brazil, and have had a great opportunity to sometimes be a part of the University of São Paulo, or UPSI. And so we call it my cell, which is Portuguese for mother cell. My is actually a word that is for mother, very similar in a lot of different languages, showing our shared history or ancestry. And so I just wanted at the end bring to attention what Dominic's already mentioned and other speakers. About 2 years ago, we came out with a medical textbook called Hematopoietic Stem Cell Transplantation Cellular Therapies for Autoimmune Diseases. And then most recently, I came out with a lay book, Everyday Miracles. It's kind of a profiles and courage of patients that have had transplant for diseases like MS, neuromyelitis optica, CIDP, Crohn's, and systemic sclerosis, as well as how it was developed, and in the final chapter, why it just has trouble taking off. And I never criticize people, I criticize systems, because that's why it has trouble taking off, is because of the system we've gotten locked into, controlled by big pharma money. In any event, I was very fortunate that Dalai Lama did the foreword for this. This book will be a trilogy, this set of lay books, and I did it because patients were always saying to me, why doesn't anybody know about this? You know, I have my longest patient with MS that's been in remission ever since the transplant, got better, stayed better, no more drugs, is 24 years. But I have patients, 10, 15, 18, 20 years, and they're all saying, why doesn't anybody know about this? So I wrote this to inform people. They don't know because I don't have a drug company behind me, and drug companies send their doctors out there that get their publications that are your currency to get promoted in academia to talk everywhere. So it's just me and a few other determined people like Dominic talking against tens of thousands of doctors, so our voice is drowned out. But it really does work, and patients are frustrated why it doesn't take off. So the next book is called Kill Switch. It's written, it's at the publisher, it'll be out this year. And that's going to be, I think, I'm even more excited about it because it really talks about molecular biology, but what it does is I give the historical background, and I'm convinced from teaching is why students, beginning in high school, unfortunately, are turned off by science or mathematics is because they're just told a concept and they're like, well, you just pulled this out of thin air, it makes no sense, and these bright minds shut down. But if you put the historical perspective in how we got to where we are, it really does make sense and it becomes very interesting. So that book will be out this year. So thank you.
Thank you. So great talk. We're all dreaming. We need your IPS soon, but maybe we could have discussion at lunchtime, if you don't mind, unless there is one burning question. How does it work? I think we have the answer. James, you want... Oh, one question over there? Yes,
Price? The price?
The price? Oh, well, you know, when we do this, you know, the phase 1, phase 2, phase 3 are free. After that, the price will be determined. And there's a board, you know, that will be involved in this. Now, from my perspective, and I don't think investors like hearing this, but I've been upfront, I will fight to make this as inexpensive as possible to help everyone around the world. I do not, the last thing, and I won't allow, let me put the caveat in, if this works in humans, I will not, absolutely not, let it be reserved for the wealthy. So, you know, one of the ways around that is we have another patent for cosmetics, and that's where people will pay money, but that money will supplement the use to help people with diseases, so they'll be able to afford it. So we've kind of thought ahead about the price. But yeah, unfortunately, doing these things costs money. You need investment. Investors don't want to put in unless they see a golden rainbow at the end of these efforts, which I understand.
Thank you.
So thanks again, Richard. And